Okay, we can start, I guess.
Okay, so shall we start? So welcome everyone. I am Fabien Balm, CEO of Biotech One. At Biotech One, we develop clean, competitive, and scalable solutions based on supercritical CO2. And today, I am pleased to introduce EncapX, our patented platform redefining nanoencapsulation. Before we begin, please share your expectations and challenges in the chat so we can tailor the discussion. And you can also book on the button a one-to-one meeting using the button. Feel free to post your questions anytime. We will address them during the live Q&A after 20 minutes presentation. And now I would also like to introduce Professor Elisabeth Badens and Dr. Associate Professor Christelle Crampon, who will be involved in to our academic partners who can answer to the questions at the end. And the speaker will be Elisandro Klein. So Elisandro, I'll let you start the presentation.
Yes, thank you, Fabien. Good afternoon, everyone. And thank you for joining us today. So today, I will present how supercritical CO2 can be used to produce continuous and scalable process for nanoliposomes and LNPs. With the partnership with academics, as Fabien mentioned, Professor Adil Moahid and Elisabeth Badens from the Aix-Marseille University. So why nanomedicine is so important nowadays? Nowadays we have several different applications for nanoparticles, going from life-saving RNA therapeutics to premium dermatology. So there is a huge interest recent on the development of new vaccines, the RNA therapies, and also oncology, new treatments. And also, most recently, there is a lot of interest from the cosmetic industry for producing premium cosmetic using active encapsulated nanoparticles of active ingredients for cosmetics. However, we still face some challenges on the industry. So we can have very good formulations for different applications, but still there is some limitations on scaling up the process. Some applications work very well on lab scale, but then when you go to the factory, to the industrial production, you have some challenges. And the gap is mainly producing continuously with good reproducibility and at large scale, mainly for high quality RNA therapeutics, lipid-based oncologic medicaments, drugs, and advanced cosmetics as well. So the conventional tools works very well at lab scale and sometimes also works on the industrial scale, but there is still some challenges and some limitations on the application of different approaches for nanoparticles, lipid nanoparticles, and nanoliposomes production. So one of the most used process is the thin layer hydration, which normally requires additional steps after the particle formation. So for example, sonication and extrusion to reduce the particle size and prepare uniform particles, which is very labor intensive and is a batched process, it's not a continuous process. That it's used currently at industry scale, but as I mentioned, there is some limitations to obtain a unimodal particle size. Mostly you obtain bimodal particle size or a wider particle size distribution. Microfluidics is also a great technology. It's used worldwide for different applications and also for encapsulation in nanoliposomes, for example. But when you go to industrial scale, there is more challenges because it's known that the process have some clogging issues, considering the lipid formulation that sometimes is more viscous. And when you go for industrial scale, you number up the number of microchannels, which also increase the system complexity that can.
Are you back, Alice?
Okay. Yeah, I think I'm back.
Yeah.
Sorry, just technical issues. And also another method is the solvent injection that normally use some organic solvents that is not ideal for sensitive APIs and also requires an additional solvent removal step. So that's why we decided to fill this gap, to find a solution for some challenges that the industry face at the moment. So we developed the EncapX, which is the first continuous supercritical CO2 system, which works on a steady state continuous operation, clog-free, operational, because it doesn't use microtubes. Instead, it uses millifluidic tubes, which is 1,000 larger than the tubes used on the microfluidics, with a very high precise, reproducible, and unimodal particle size distribution. Very versatile to produce nanoliposomes, lipid nanoparticles, and other lipid-based particles, compatible with mRNA, siRNA, and diverse other lipids and active compounds for drugs and for cosmetics and food tech also. Mainly using supercritical CO2 as a solvent, which is a green solvent. No organic toxic solvent is used on the process. So why supercritical CO2? So supercritical CO2 is a specific state of the gas, the CO2, where we can achieve specific conditions and, for example, under pressure and under specific pressure and temperature, the CO2 acquires a liquid-like density, which improves the solvating capacity and modifies the interfacial properties. It also has very low viscosity, which enables efficient fluid mixing and formation of a CO2 in water or water and CO2 emulsion under pressure, depending on the formulation that you have. So the mechanism inside of the process involves the CO2 in the supercritical state, which interacts with the aqueous, ethanol, and lipid phase, reducing interfacial tension and promoting the formation of a homogeneous pressurized system. And the nanoparticles are formed during the controlled depressurization when the CO2 is released. So you have a thermodynamic self-assembly triggered by depressurization, and you don't have like shear base size reduction steps that sometimes are used to form particles, nanoparticles. So just a simplified vision of the process. So it's a continuous process where you have two streams, two enter streams, one with the lipids, the API, the phospholipids, and water and ethanol. And then you have also the supercritical CO2. We have this millifluidic mixing device, which is our patent process, our patent device, that produces under pressure the emulsions of CO2 and water and CO2 with very fast mixing inside, and is a process without any autoclave. So for those that are familiar with supercritical devices, normally you need to have an autoclave. And our main innovation here is that we don't have autoclave. And the autoclave is one of the most important costs on the implementation and operation of supercritical processes. So we have just tubings and all the process, of course, inside of the tubings without an autoclave. So it's very innovative approach for supercritical CO2 processes. Because as far as we know, there is no other application for supercritical CO2 without an autoclave. And at the end of the process, where you have the depressurization of the mixture, the CO2 is released automatically, instantaneously, and you have the formation spontaneously assembly of the nanoparticles. And with this process, you can also recycle the CO2. So it's a very green process, very eco-friendly process. And as I mentioned, it's a continuous process. So you have continuously the two enter streams and the exit and collection of the nanoparticles. So what kind of results you can achieve with this technology, with the EncapX? We can achieve a very narrow particle size distribution with a low PDI, which increase the stability of the formulation, and we have a precise control on the particle size formation. It's very reproducible, so every time that you use, if you use the same formulation, you can control the operational conditions to achieve every time the same particle size and the particle size distribution. And you have a unimodal particle size, which means that you have just one range of particles. You don't have agglomerates or big particles, which also leads to a higher stability and also improve the bioavailability of the compounds. We can achieve very high encapsulation efficiency, both for hydrophilic and lipophilic compounds because of the nature of the phospholipids that we use in the formulation. And it's a very robust and user-friendly process and equipment that's very stable, very low maintenance, that works very stable and reproducible between different runs in a continuous mode. And it's very easy to use process with no difficult parts to operate and to develop new formulations using this equipment. So here just a case study where we developed the encapsulation of an anti-cancer drug called docetaxel. Docetaxel has some some challenges for the application. It's a very important oncologic anti-cancer drug, but there is some limitations on the solubility and on the stability of the compound. So the encapsulation that we performed using the EncapX system produced a unimodal particle size distribution around 90 to 120 nanometers with a very gentle temperature around this one was with 35 degrees. So it's very gentle, which is important for sensitive ingredients, with an encapsulation efficiency higher than 95% and a consistent performance in continuous mode between runs. So it demonstrates the importance and the viability of the process for industrial application with oncology difficult ingredients. Today, we are focusing on the anti-cancer drug, but we also have some validated results and ongoing projects with encapsulation of different kinds of RNA, and also for different active ingredients for cosmetics and for food tech. But we can talk a little bit more about that on the questions or on the next webinar that we are planning to organize in a few months. So yes, the application is very wide for this technology. So we can prepare nanoparticles for pharmaceuticals like RNA encapsulation, oncology drugs, peptides, and controlled release delivery systems, because it's one of the properties of the nanoliposomes. Also for premium cosmetics, like retinol, antioxidants, botanical extracts, and improving the dermal absorption because of the particle size, which is compatible also with the Clean Beauty label. That's very good for cosmetics because it doesn't use any toxic solvents or any rush process, using CO2 that's completely removed after the process. And it's also, of course, applicable for some food tech applications like aroma encapsulation, natural antimicrobials, and nutrient protection, because it's widely known that nanoliposomes and lipid nanoparticles can protect from light exposure, temperature changes, pH changes. So depending on the formulation, you can protect different ingredients for different applications. So just a quick summary showing the state of art at the moment, the different process for nanoparticles production, and where EncapX is positioned. So we have the thin layer hydration. It's a batch process with low control on the scaling up, demanding additional steps to reduce the particle size. And often it uses toxic organic solvents, which is a problem for some applications that demands to remove the residual solvent, or sometimes you are not able to remove everything. So it's a limited application depending on the formulation. Microfluidics, as I mentioned, works very well at lab scale. You can have great formulations, but to scale it up is a challenging process. So sometimes you are not able to scale depending on your process because you have some clogging issues. The solvent mixing process also work as batch or continuous, but have a moderate ability to scaling up, and also often use organic solvents. And EncapX came as a new option using a totally continuous process with very high capacity to scaling up. We also perform the scaling up, numbering up the number of the millifluidic mixing devices. But as we use a millifluidic tube instead of a microfluidic, we don't have clogging issues, and the scale is by itself higher because of the size of the tubes. And we don't use any kind of organic toxic solvents. We just use the ethanol on the formulation, which is very normal for different applications, that is easily removed after the particle formation. So Biotech One have the patent IP for this process, for this equipment. We have, as I mentioned before, validation for different active ingredients, from anti-cancer drugs to RNA and some cosmetic ingredients. And the process is GMP ready. There's a concept GMP ready, and we can also offer industrial support for our customers. So at Biotech One, we have a deep knowledge on the high pressure engineering supercritical CO2 mainly for different applications. We have experts, top experts on our scientific board also. And we have a good knowledge on lipid science and process design, also always thinking about how to have an efficient process that can be used at industry scale, because you can have some good process at lab scale, but it's very important to have it also available for industrial production. And during the last couple of years, we have been collaborating with different academic institutions, industrial partners in different sectors, from pharma, cosmetics, food techs, and so on, with different validation of the process. So we have the continuous control, scalable, and ready process for the next generation. And the question that I leave here is, what would you like to encapsulate with us? And I will open also the question and answer section. Feel free to ask. And also, as Fabian mentioned, you can click on the link to book a meeting directly with us, where we can discuss in more details how our technology can collaborate with you.
So we already tried. Thank you very much, Alessandro. We already tried to answer to some questions on the chat. As you can see, we can also talk about that. And we are pleased to welcome our academic partners, Professor Badens and Dr. Adil Moaid. So they are the inventors of the patented process. Their scientific expertise has been essential to the development of this platform. So now you can go to the question on the chat.
Yes, I can see the question
directly. And of course, if there is some confidential points, don't hesitate, book a meeting, and we'll talk privately.
Okay, yes, just answering a question about the pressure required for CO2 to make proper liposomes. The CO2 goes to the supercritical state after above around 74 bar. But we validated most of the process using 150 bar, which also depend on the formulation that you have and the results that you want. But yes, inside of the equipment, the active compounds will not be at, how can I say, a harsh condition. It's not directly related to the pressure. Also a question, how to prevent agglomeration. So as the process is a very fast mixing and a very fast depressurization, we don't have agglomeration problem during the particle formation. So it is produced as a very fast jet where you collect the liposomes, and we collect in a very dispersed way. So there is no problem for agglomeration. Okay. Yes, if there is some need and guidance to achieve higher encapsulation, you can schedule a meeting with us so we can discuss in more details how we perform the encapsulation to achieve this high encapsulation efficiency. But I can say that it's also related with the technology that we have. Okay, let me see. Okay. Yes. Okay. Yes, I think that now I will not be able to show the picture of the equipment, but just to give you an idea about the size, is a bench size, is very compact, very small equipment. I will say like you will need like 1 meter in your bench, on your lab bench to have it. and is everything inside, I will say, like easy to use equipment, like a box. It's totally automated, so you can just select a preset operational conditions that we can optimize before the use, or you can also do the optimization during our R&D process. And then you just need to click one button, and the equipment will do all the next steps. And when the encapsulation is completed, the equipment stops, and you just need to collect the encapsulated material. So it's very user friendly, very easy to use. Because we know that sometimes people hear like supercritical CO2 and think that's something very complicated and difficult to use. And we make sure that this application is very user-friendly and it's not complicated. You can use it with a very short training, and it's very easy. So let me see. Yes, the characterization of the nanoliposomes and the lipid nanoparticles we do using DLS. We are also exploring other technologies for characterization, but as far as now, we have achieving good results using the DLS. And of course, if you want to see more specific details, you can ask by email or booking a meeting, and we can share some non-confidential results. Because we have also some limitations in some results that we can share, because we do some projects with confidential contracts with companies. So we have limitations in some data to share, but of course we have some non-confidential confidential information to share. If you want to see more, you can book a meeting with us or send an email. Let me see, key components to make nanoliposomes. Yes, the stability of the nanoliposomes will be mostly related with the narrow particle size distribution and the unimodal particle size. And it will also depend on the formulation to increase the stability, but having a narrow particle size distribution and unimodal particle size will reduce the agglomeration and so on. So yes, can you share this information with us? Yes, we have images for the nanoparticles. Yes, I forgot to put in the presentation, but we have images with TEM, not with SEM, we have with TAM, that we can see clearly the bilayer of the particles and the unimodal particle size distribution, but you can contact us and we can also share the images of the particles on the microscope, of course. They mentioned that no organic solvent are used, so that means that lipids particle needs to be soluble in water. No, actually, there is a question about the lipids or compounds to need to be soluble in water. That's actually the advantage of producing nanoliposomes, that we can encapsulate both lipid-soluble and water-soluble compounds. We can encapsulate it on the nanoparticles, and then we have this suspension of nanoparticles in water. So if you have limitations of solubility of lipid-soluble ingredients, for example, that you cannot use in your formulations, you can encapsulate using nanoliposomes, and then these nanoliposomes will be stable in water suspension. So that's the case of the docetaxel, for example, because there is a very low water solubility of the drug. And with the encapsulation, we can increase the bioavailability and the stability of the compound in a water suspension. So If I'm not clear, you can ask more informations also. Let me see what else. Okay. Okay, yes, I think that was all the questions on the chat. If anyone has any other questions, you can ask. Yes, for the docetaxel formulation, we used some phospholipids and cholesterol. I'm not sure. You can schedule, please schedule a meeting with us so we can discuss in more details about the formulation, because I will need to check how far I can go disclosing the specific formulation. But with meeting, I think we will be able to disclose more details than here. Other solubility, I want to know in which media you inject the lipids. Okay. One question about in which media we inject the lipids in API. So when you have the two flows, I can go back here. So you have the two flows, you have the flow of supercritical CO2 and you have the flow for the lipids and the API solution. In that stream, you need to, if it's not water soluble, you need to solubilize in a mixture of ethanol water. So we normally use a mixture of 29% ethanol and 71% water, which is able to solubilize the lipids, the phospholipids, and the active lipophilic compounds. And then at the end, of course, you can remove the remaining ethanol using quick process like evaporation or dialysis and so on. Yes. Any other question? Yeah, so everyone feel free to reach us by email, in our website or our LinkedIn. And also feel free to click on the call to action to book a meeting. And I think
There's another question.
no more question, I think. Okay.
Nanoparticles.
Okay, I'll determine nanoparticles that are not lipid-based, polymetric nanoparticles, for example. Yes, in theory, we are able to produce also polymeric nanoparticles, but we didn't validate the equipment for that yet. But it is on our radar for later validation on the next months to develop different applications for different nanoparticles as well. But until now, we didn't validate it for that, mainly for lipid-based nanoparticles. Solvent-free liquid nanoliposome was your solution. Yes, with our equipment, you can produce solvent-free liquid liposomes. So you can have it just a suspension of nanoliposomes in water, certainly without any toxic organic solvents, which is very good for clean label formulations.
And because most of the studies are confidential studies, so don't hesitate. Of course, we sign an NDA, and we design a ready-to-market equipment, which is EncapX. But above all, we are a research and development company. So we are here to support you, our partners, through feasibility studies, proof of concept project, and validate your formulations in order to de-risk and scale up and accelerate from lab scale to industrial deployment. So don't hesitate. We can perform proof of concept optimization studies. And
yeah, so for example, if you have
Or you
If
can
you
buy
have already some formulation that you're using another kind of encapsulation process, you can contact us and we can perform a proof of concept with the same formulation using our technology, so you can see the difference on the results. Yeah, any other questions? Yeah, I think we don't have any other questions for now, but feel free to contact us.
So to conclude, our objective today was to show that nano-encapsulation can be cleaner, more precise, and truly scalable. If what you've seen today resonate with your challenges, we would be very pleased to continue the discussion. So thank you very much for your attention. If there is no other questions, we will conclude. Of course, we'll. A lot of people asked to participate, but they couldn't. So we'll send a short presentation, but we'll have other presentation in the next
Yes,
week.
and keep connected in our LinkedIn, because we will organize other webinars on the next months also.
With other specific case studies and other application like extraction or so.
So thank you, everyone.
Thank you very much for your attention, and goodbye, everyone. We hope to see you. Goodbye. Someone is one, Mr. Kovidanchari. Open the camera. Do you want to speak? No, no question. Okay.
Okay. So
Thank you very much, everybody.
thank you. Switch off the meeting.
Okay.